I'm Emily Dolson. I'm currently a postdoc at Cleveland Clinic, and I'm going to be talking about some of the latest work that we've been doing on controlling the speed of evolution. One of the long-term goals for gaining a really deep, solid understanding of evolutionary theory is that if we can do it well enough, we might be able to control the long-term trajectories of the evolution of bacteria and cancer and stop them from evolving resistance to our treatments. And we're actually already starting to make some progress on that by finding ways of sequencing drugs that can kind of trick the pathogen through the part of the evolutionary landscape that won't lead to the evolution of resistance. But thus far, there's been a really serious obstacle to that, which is the fact that we don't know how long we need to apply each drug for in order to guarantee that the population is at the part of the fitness landscape that we need it to be in order to have the outcome that we want. We set out to derive some rigorous mathematical theory for controlling the speed at which evolution progresses in these contexts. And we actually found a way to do it, assuming you have a few pieces of preliminary information. First, you need to know all of your kind of standard evolutionary parameters: mutation rate, population size, etc. Although actually some preliminary work suggests you can be pretty wrong on some of those and it will be okay. Second, you need perfect knowledge of all of the relevant parts of not just one fitness landscape, but an entire sequence of fitness landscapes that exist along some sort of environmental change axis. These do need to be fitness landscapes that you are capable of subjecting your population to. And third, you need a way to sequence these fitness landscapes such that if you had infinite time, you could get your population to your desired endpoint. We're still nailing down exactly what properties the environmental change axis needs to have in order to make this work, but there probably are some. So the math that we used to do this was borrowed from a technique that was developed a couple years ago in quantum mechanics called counterdiabatic driving. The problem that counterdiabatic driving is designed to solve is you have a noisy system and you want to move it from one position to another. And so originally this was some noisy quantum system, but in our context, it's a population. We want to move it from one point in the fitness landscape to another. So normally there's a trade-off when you're doing that between how quickly you can move and how precisely you can control the system while you're moving. My favorite metaphor for thinking about this comes from this one paper, which described this system as being kind of like a waiter carrying a glass of liquid on a tray. If the waiter holds the tray perfectly flat and tries to move the liquid, it's going to slosh around because there's going to be air resistance and so on that'll perturb the system. So one way that the waiter could get the drink to a person's table would be to move infinitely slowly. However, waiters, just like doctors, do not actually have infinite time. What waiters actually do is they move very quickly, but they tilt the tray as they're moving in a way that compensates for the air resistance and all of the other factors that will perturb the glass. Counterdiabatic driving works the same way. Rather than traveling between a start state and an end state via a sequence of states that you know will induce noise via perturbations, you can travel between the same start and end state using a different sequence of intermediate states that you've pre-calculated to compensate for those perturbations. If you had used the original sequence of states, the noise would mean that your system would take time, perhaps infinite time, to equilibrate to each of those new states. A perfect counterdiabatic driving protocol will completely eliminate this lag. To see if this actually works when we translate it to evolution, we tested a counterdiabatic driving protocol on an empirically derived 16 genotype fitness landscape from malaria resistance genes. And of course, it's not just one fitness landscape. It's a sequence of fitness landscapes for different concentrations of an anti-malarial drug. So we selected a sequence of drug concentrations that produced a lag between the observed distribution of genotypes in the population and the distribution that we would expect to see at equilibrium for each of the drug levels. To be clear, this is a digital population. We're working on testing this in the wet lab, but we're not there yet. And when we compared the distributions of genotypes that we observed under the counterdiabatic driving protocol to the distributions that we would expect at the equilibrium for all of the different drug doses in the original protocol, we found a very close match. So the counterdiabatic driving protocol does indeed eliminate the lag. It doesn't eliminate 100% of the lag because this isn't an idealized perfect counterdiabatic driving solution, but it eliminates enough of it to be useful in practice. So ultimately, we think this is a pretty good step forward towards being able to actually, in practice, control evolution in the real world. Like I said, this was a huge multidisciplinary collaborative project, so I would really like to thank all of my many co-authors and thank all of you for listening.